Okay, so welcome to this next video in which we are discussing the palmitoylation of proteins and palmitoylation inhibitors. Okay, right, so we've now discussed the two main divisions of palmitoylation. We've discussed N-palmitoylation and S-palmitoylation of proteins, and I now want to give you some examples of when cysteine residues will be S-palmitoylated in proteins, because as I said in the previous video, there are a huge number of different cysteine residues within the polypeptide. Not all of them are going to end up being palmitoylated. So what actually determines whether a cysteine gets palmitoylated? When will a cysteine be palmitoylated, basically? So firstly, I should just say the answer to this question is not known, basically. However, all we can say is that there are certain circumstances in which palmitoylation seems to occur. We don't understand how these circumstances lead to the palmitoylation, however, but we know certain circumstances in which palmitoylation does occur. Okay, so the first one to mention is if the cysteine residue is within or near a transmembrane region. Okay, so what do I mean by this? Well, basically, in proteins which are within membranes, i.e. which straddle lipid bilayers, basically, they have these alpha helices which straddle the membrane. So let me just draw the phospholipid bilayer here. Okay, and now let's draw a protein going through this. So basically you have an alpha helix like so, which just goes through the membrane. Now if you have cysteine residues within that alpha helix, those tend to get palmitoylated. And this makes sense because if you palmitoylate them, what will happen? You'll have a nice big palmitoyl group sticking off the side of the alpha helix, and this will get to interact with the hydrophobic core of the lipid bilayer. And of course we've seen palmitic acid. It's an incredibly saturated molecule, therefore it's incredibly hydrophobic. Okay, so it will interact beautifully with the hydrophobic core of the lipid bilayer. Okay, so cysteine residues within transmembrane domains or near to transmembrane domains. So for instance, if you had a cysteine residue up here, it might end up getting palmitoylated, and the palmitoyl group would then implant itself into the phospholipid bilayer, and that would stabilize the protein within the membrane, basically. So that's one scenario in which we see palmitoylation of cysteine residues. Okay, let me go on to the second one now. Now this is more complicated. That one was nice and easy to explain. This is more complicated. In order to understand this, we are going to have to talk about a different type of lipid modification that occurs to proteins, namely prenylation. Okay, so let's take on the topic of prenylation. Okay, so basically, proteins which are going to be prenylated, which is another type of lipid modification that you can make to proteins, and I'll explain exactly what a prenyl group is in a moment. They have a special structure at their carboxylic acid terminus. Okay, so let's say this is the start of the polypeptide, this is the amino terminus, and this is the carboxy terminus. So why don't I just draw those things? Basically, if you are going to get prenylated, you have to have a certain structure at the end of your polypeptide, okay? So you have to have what's known as a CAX box, okay? And proteins which do have a CAX box are called CAX box proteins. Okay, now what does this mean? Well, it means that the final four amino acids, so let me just draw these in, 1, 2, 3, 4. The final four amino acids have to have certain properties. So the fourth from last amino acid has to be a cysteine. That's what the C here is for. The third from last amino acid has to be A. Now that does not mean alanine. If you wanted it to be an alanine, you would put a capital A. Instead, little a like that means that it needs to be an aliphatic amino acid. Okay, so let me give you some examples of aliphatic amino acids. And basically, aliphatic just means well, it means non-aromatic, firstly, and also it tends to imply hydrophobic as well, so no fancy functional groups. It basically just means that you've got carbons and hydrogens in your R group. Okay, so let me give you some examples of aliphatic amino acids. Now, I won't draw out the 4 amino acid structures, I'll just draw out their R groups, so the variable portion. Okay, so glycine is an aliphatic amino acid. It's ridiculously simple, but it is an aliphatic amino acid. Its R group is just a hydrogen atom. Alanine is an aliphatic amino acid. Okay, its R group is a methyl group. Another example is valine. Valine is starting to get a little bit more complicated. In valine, what you have is a carbon, which then has 2 methyl groups coming off it. So 1, 2. So that's the R group of valine. Then we're getting bigger as we go down. Leucine is another example of an aliphatic amino acid. Now, leucine is effectively just a bigger version of valine. Okay, so it's basically just valine with an extra methylene group put in. Okay, so here's the methylene group. Now just stick the rest of valine on here. So a carbon with 2 methyl groups sticking off it. That's a leucine. Then the final example of an aliphatic amino acid is isoleucine. Okay, so let me, oh no, actually it's fine where it is. Isoleucine, what you do is you move this hydrogen, and, well, what you do is you swap one of these hydrogens for one of these methyl groups, so effectively swap this round and put one of these methyl groups onto this first carbon here. Okay, so it's a structural isomer of leucine, hence isoleucine. Right, and then we have the methylene group now here, and then a methyl group coming off there. Okay, so that's the structure of isoleucine. So those are 5 examples of aliphatic amino acids which you could have in this position, third amino acid from the final one. Okay, then again in the second amino acid from the final one, again you need an aliphatic amino acid. And then finally, in this final position, X. Now, X doesn't mean any amino acid. No amino acid has single letter amino acid code X. Okay. However, this final amino acid will determine which prenyl group we're going to add on, and I will explain what a prenyl group is in a moment. So let me just do this bit and then I'll explain what the prenyl groups are. So basically, if in position X you have an alanine amino acid, which we've already seen has a methyl group as its R group, or you have a methionine amino acid, which we saw earlier was the initiator amino acid, but it can be incorporated later on as well. Okay, and the R group of methionine is an ethylene group, which basically just means 2 methylene groups joined together, then with a sulfur atom, and then a methyl group on the end here. So that's the R group of methionine. Okay, and then finally, if you have a serine amino acid in position X, okay, all of these are going to give rise to the same prenyl group being added on. Okay, so basically, if you want to be prenylated, you have to have one of these CAX box structures, and what it means is that we're going to stick a prenyl group onto this cysteine, which is 4 amino acids from the last, that's in the CAX box. Now, if in position X you have an alanine, a methionine, or a serine amino acid, and I didn't get around to completing the R group of a serine amino acid, but it's a methylene group with an alcohol group off, okay? If you have one of these, you are going to add something known as a farnesyl group. So all of these will lead to a farnesyl group being added onto the cysteine residue. Okay, and I'll explain exactly what that is in a moment. So there are only 2 prenyl groups that can be added onto the cysteine residue. Whew. One of them is farnesyl, and if you have an alanine, a methionine, or a serine in position X, that will promote you having a farnesyl group added on. However, if your if your X amino acid is a leucine, which we've seen over here, you will instead get the other prenyl group added on, which is a geranyl prenyl group. Okay, so now that I've explained the CAX box, I'll move on to explaining the prenyl groups and what will happen after you've added the prenyl group onto the cysteine residue within the CAX box. Okay, right, so farnesyl and farnesyl and geranyl. Okay, so these two things here, these are what are known as prenyl groups. Okay, so they are not molecules, they are groups. You add them onto molecules. Just like this is not a group, just like this is not a molecule, it's a group because it needs to be added onto some other molecule. Farnesyl and geranyl will have one of these basically. They'll have a bond that needs to be attached onto something. Okay, so they are what are known as prenyl groups. Now, why are they called prenyl groups? Well, basically it's because they are based on a molecule known as isoprene. Okay, so let me show you the structure of isoprene, and then I'll show you how you can get farnesyl and geranyl groups from isoprene. Okay, so I will draw the skeletal structure of isoprene, because when we're drawing farnesyl and geranyl, it just gets messy if you don't draw the skeletal structure. Okay, so we might as well get used to it. So, isoprene just consists of carbon atoms and hydrogen atoms, which means that its skeletal structure is incredibly simple. Okay, because we don't show carbon atoms and we don't show hydrogen atoms attached to carbon atoms. So, we have 5 carbon atoms here. 1, 2, 3, 4, and then a 5th one up here. And wherever there are missing bonds, that will be because you have hydrogens coming off those carbons. Okay, so we have 2 double bonds, 1 here and 1 here. Okay, right, so what is now going to happen? And I'm just going to actually quickly draw the molecular formula just so that this is fully clear. Okay, so here is the molecular formula for isoprene. So let's just add on the hydrogen there and add on 2 hydrogens here. Okay, because it will make it clearer for when we're trying to actually, when I'm trying to explain what's about to happen. Okay, so basically we want to construct farnesyl and geranyl groups out of isoprene. So how do we do it? Well, basically you can polymerize isoprene together, and farnesyl will be when you polymerize 3 isoprene molecules together, and geranyl will be when you polymerize 4 isoprene molecules together. Okay, so let me try and explain this. Imagine breaking the second of these 2 bonds the second of the 2 bonds in both of the double bonds, basically. So don't break both bonds in these double bonds, just break the second of the 2 bonds. Now, double bonds are just like normal bonds. Each of the 2 bonds in the double bond has 2 electrons in, so when we break one of the bonds, that will mean that we need to return 2 electrons to the components. So imagine returning 1 electron to this carbon and 1 to this carbon. Do the same for this bond breaking. Return 1 electron to this carbon and 1 to this carbon. Okay, now what we'll do is we'll bind this carbon to this carbon because they both now have 3 electrons. So let me show you this kind of imaginary intermediate because it will help us to understand what's about to happen. Okay, so we form this double bond between the 2 carbons in the middle, and now we've got single bonds to these methylene groups, and of course both of these carbons on the end still have 3 electrons. Okay, so I'll demarcate this by putting big pink dots to represent the 3 electrons there. So of course this isn't a molecule, this is just our imaginary intermediate here. Now let me just draw you a skeletal formula for this, because when I start showing farnesyl and geranyl, I'm not going to bother drawing out the full molecular structure. So a skeletal formula for this imaginary intermediate would be like so. We've now got a double bond between these 2 middle carbons, and of course we need to remember that these 2 end carbons have those 3 electrons. Okay, now what you're going to do basically is you're going to get multiple of these. So for instance, let me bring along another one here. Okay, so here is another one of these isoprene intermediates here, and it will be in exactly the same situation. It will have a free electron here and a free electron here, and what we're going to do is we're going to bind this one, well, this carbon here, to this carbon here, which we can do because both of them have free electrons, and that's how we're going to polymerize these isoprene molecules together. So you can imagine you could do this for, ever, basically. Now, we're not going to quite do it forever. Instead, we're going to do it 3 times for farnesyl and 4 times for geranyl. So we're almost there, basically. Now let's just add one more in for farnesyl. Okay, so here is our third one, and again, we'll have a free electron here and a free electron here, and what we'll do is we'll bind this carbon to this carbon here, and that now will nearly be a farnesyl group. Now, you'll notice there's a problem here. Okay, we have a free electron here and a free electron here, so we'll bind the free electron on this carbon to some unknown group as of yet, some unknown molecule. Okay, so that's why this is called a farnesyl group, because we don't know what this is going to bind to, basically. So this is the unknown bit. And you might be wondering, well, what about this side? Well, we're just going to bind a hydrogen to that, basically. So just bring a hydrogen atom and bind it to that final carbon over here to make it a methyl group, basically, and that's happy. Okay, right. So this, which I've just drawn for you here, is a farnesyl group. Now, if you did it 4 times, which I'll do in a moment, that will create you a geranyl group. Now, before I show you the structure of geranyl group, I want to show you how we're going to add this farnesyl group onto a cysteine residue. Okay, so let's get our cysteine residue back again. So here is an amino group, and remember, we're specifically adding it onto this cysteine residue that is at position 4 in from the terminal amino acid within the CAX box. Okay, so we know exactly which cysteine we're adding this onto. Okay, so here it is. Here's the amino group, here's the alpha carbon, the carboxylic acid group, and then the R group of that cysteine residue within the CAX box consists of a methylene group, then with a thiol group here. Now, to prenylate that cysteine residue, what you're going to do is you're going to cleave this bond between the sulfur atom and the hydrogen atom of the thiol group of the cysteine residue. And again, you can imagine giving one of the electrons back to the sulfur atom and one back to the hydrogen atom. This will create you a hydrogen atom, which you can imagine is the hydrogen atom that you would use to bind in over there. And then you would attach this sulfur atom onto this carbon atom, because both of them now would have free electrons. So this is how you're going to attach this farnesyl group onto the cysteine residue. Okay, and you would now say that this cysteine residue has been farnesylated. Okay, so this is farnesylation. Okay, so it's farnesylation. Okay, right. And this type of bond that you have here, okay, now, remember I said sulfur is very, very similar to oxygen. What would you call this sort of bond if it was an oxygen? If you've got an oxygen sitting between 2 carbons, that's called an ether link between the 2 carbons. Okay, so when you've got a sulfur between 2 carbons, as we have here, that is called a thioether link. Okay, so the farnesyl group is attached to the cysteine residue via a thioether link. Okay, so that's nice. I'll just put the link in there. Right, so that's farnesylation, and farnesylation is a form of prenylation. So prenylation just means adding prenyl groups on. Farnesylation is a form of prenylation because a farnesyl group is a form of a prenyl group. Now, the other form of prenylation which occurs biologically is geranyl-geranylation. Okay, so let's do this just so that we are fundamentally clear, but it's exactly the same process. All that will change is that you'll add on a final isoprene molecule over here. Okay, but just to revise, basically, because this is a difficult thing to take in for the first time, so we might as well revise. Okay, so let's do geranyl-geranylation. So, basically geranyl groups are going to consist of these, they're going to consist of these 4 separate isoprene intermediates. So let's draw one of these out. Okay, I'm just leaving space for the cysteine residue that I know we're going to have to draw later. Okay, so here is one of these isoprene intermediates, and I won't put the pink dots this time because it makes it look kind of messy, but we should remember that both of these carbons have 3 electrons. Okay, now I'll draw the second isoprene intermediate here, and the third here, okay, and the fourth over here. Okay, and then what we're going to do is link this one up to this one up to this one, this one up to this one, put a hydrogen off that, and we're going to attach this carbon onto the sulfur atom, and this then is called a geranyl group. Okay, right, so it's 4 isoprene molecules polymerized together, and we're now going to add this to the sulfur atom, so here's the sulfur atom, of our cysteine residue here, and remember the cysteine residue will be within the CAX box, so it will be the 4th cysteine residue from the end of the polypeptide. Okay, and then here is the amino group over here. Okay, right, and you would say that the cysteine residue has been geranylated, or you would call this process gerynylation. Right, okay, so you can add these prenyl groups onto the cysteine residue of proteins which have CAX boxes. And as I said, there are certain amino acids that you need to have in the X position to have each of these prenyl groups put on. So to have a geranyl group put on, you need leucine, remember, and to have farnesyl put on, you need alanine, methionine, or serine in that X position. Okay, right. Now, let me just mention something called post-prenylation modification. Okay, so let me just draw my protein back again here. And let's now say we have our cysteine here, we have our CAX box, and let's say prenylation has occurred, so you've got some prenyl groups sticking off here. What then follows is the protein then has the AAX terminal free amino acids removed. So what follows is a process known as AAX proteolysis. Okay, so basically what happens is you cut those final 3 amino acids off of the polypeptide. So all of this is taken off, and the cysteine residue with the prenyl group stuck on becomes the final amino acid of the polypeptide. So this will basically become this. Okay, and cysteine is now the final amino acid with this prenyl group stuck on. Then what happens is you methylate the cysteine residue. Okay, so these are the 2 post-prenylation modifications. Firstly, AAX proteolysis occurs, and then methylation. Okay, so I'll just write their titles, and then I'll show you what methylation is all about. So these are post-prenylation modifications, and I don't think modifications is going to fit in, so I'll just put post-prenylation there. Right, so let's draw out the structure of our final cysteine residue now. So here it is again. So here's the amino group, here's the alpha carbon with its hydrogen coming off it, and its R group, which is this methylene group with a sulfur atom, and then with the prenyl group stuck on, which I'll just abbreviate to prenyl now. Okay, and then finally it now has this free carboxylic acid group because it is the final amino acid of the protein, basically. Now what is going to happen is you're going to take this hydrogen off that alcohol group, and you're going to replace it with a methyl group, basically. And this is why it's called methylation. You're going to stick a methyl group onto that oxygen atom of the carboxylic acid group in place of this hydrogen. So cut this off, put this on. Okay, so that's the process of methylation, and that follows prenylation. Now, why have I told you all of this? It's fantastically interesting and all, but why is this relevant to palmitoylation? Well, basically, if you have other cysteine residues that are nearby cysteine residues which have been prenylated, so terminal cysteine residues which have been prenylated, if you've got other cysteine residues that are nearby those, maybe 2 or 3 amino acids away, then those cysteine residues will also get lipid molecules stuck onto them, but they won't get prenyl groups stuck onto them. They will get palmitoyl groups stuck onto them. So, to distinguish between a prenyl group and a palmitoyl group, I'm going to colour them in different colours. So in green we'll have the palmitoyl group, and in pink we'll have the prenyl group. Okay, so cysteine residues that are nearby cysteine residues that have been prenylated, and the only place that prenylation occurs is on these CAX boxes. Okay, so cysteine residues that are nearby terminal cysteine residues which have been prenylated will be palmitoylated, basically, and that's the second example where we find palmitoylation. So we've had transmembrane regions, we've had cysteines that are nearby terminal cysteines that have been prenylated. Now let's extend that concept. A third example, so I'll put this here, 3, is basically any cysteines that are nearby the amino terminus or the carboxylic acid terminus at all. So remember, not all proteins have a CAX box, okay? Some will not, therefore, end up having a prenyl group put onto their ends, basically. Okay. However, if you have cysteine residues near amino termini and near carboxylic acid termini, these tend to get palmitoylated. So cysteine residues near the start and finish of the protein, we recognize that these are more likely to be palmitoylated than cysteine atoms in the middle. So cysteines near terminal cysteines which have been prenylated, cysteines near the start and finish of proteins, cysteines that are in transmembrane regions. Those are the 3 places so far. There's one final thing that I need to tell you about, and this is dual acylation. Okay, dual acylation is also going to involve something called myristoylation. But I will introduce myristoylation to you in the next video.